It's time and people are there.
Waiting.
Mm. Okay. We are now, there is 80 people are watching right now. So let's start. Shoma, you start
Yes.
introductions and everything. Then Dr. Bala will be present his presentation. Then the question, then the interview, finally question answer. Okay.
You start the recording and then
Yes. Let's start, Shoma.
good to be back. So hello and welcome to Bioingene webinars. It's wonderful to get back to this platform and meet my big Bioingene family. So some of you may be new, so I'll just give a brief introduction about Bioingene. Bioingene is a non-profit organization created to promote plant science research worldwide. This is an open platform from which plant scientists can present the current advancements of their research to the world. We hope future scientists, such as yourselves, can gain perspective and inspiration, hearing these esteemed plant researchers talk about their scientific accomplishments and views on building the future of plant sciences. We are thankful to the speakers and viewers of our Bioengine family for your interest in the webinar series and for your appreciation of our efforts. You can register for these Bioengine webinars through our website, bioengine.com, or our social media pages. You are live viewing this webinar and you can watch it again later on the Bioengine YouTube channel. So please remember to subscribe so that you don't miss out on any of our events. After attending the talk today, you can apply for a certificate of participation via the feedback link which will be provided in the YouTube chat during or after the presentation. Please type in your questions related to today's talk in the YouTube chat box for the Q&A session. The topic for today's webinar is decoding the sugary secrets during plant-microbe interaction at the cell wall interface. Our esteemed guest speaker today is Dr. Balakumaran Chandrasekar. He is an assistant professor at the Department of Biological Sciences at BITS Pilani, India. His research focus at the BITS Pilani is on the glycobiology of plant-microbe interaction at the cell wall interface. He completed his bachelor's degree in biotechnology at Tamil Nadu Agricultural University, Coimbatore, and then he received a prestigious scholarship, the Scholarship Plus Award from the German government, to pursue his master's degree in plant sciences at the University of Bonn, Germany, in 2011 to 2013. He started his Ph.D. research at the Max Planck Institute for Plant Breeding Research in Germany. And as a recognized student, he has conducted his Ph.D. research at the Department of Plant Sciences, the University of Oxford, UK, from 2014 to 2017. During his PhD research, he has employed activity-based protein profiling, a chemical proteomic approach to monitor the activities of plant glycosidases during bacterial infections. His PhD research period introduced him to the world of glycobiology. An immense fascination towards the structure of glycans motivated him to pursue his postdoctoral research at the University of Düsseldorf, Germany from 2017 to 2020. During his postdoctoral research, he has employed synthetic biology approaches to produce complex cell wall polysaccharides in yeast and Arabidopsis. After his first postdoctoral period, he received the C+ postdoctoral fellow to pursue his research at the University of Cologne, Germany from 2020 to 2021. During this period, he structurally characterized and investigated the biological functions of beta-glucan fragment derived from Piriformospora indica during colonization of barley roots. In June 2021, he returned to India to join the Department of Biological Sciences at BITS Pilani. Thank you, Dr. Chandrasekar, for joining us today. The Bioengine platform is all yours now.
Okay. Thank you very much, Soma, for the kind introduction. And also, I would like to thank the Bioingene team for inviting me for this talk. And also, I think it's a very good platform. I have also seen a lot of webinars which has been conducted by Bioingene. And these webinars are really useful. And I strongly believe that this will be a very nice platform to gain a lot of knowledge, especially in the plant science and also in the other field of research. And once again, I thank Bioengine for giving me this opportunity and join today for the webinar where my title of the talk, which already Soma has told, it is about decoding the sugary secrets during plant microbe interactions at the cell wall interface. So currently, I'm assistant professor at the Department of Biological Sciences at the BITS Pilani in the Pilani campus. Today, in the webinar series, I would like to take you through the sweet journey which I had so far, right from my PhD towards until now as an assistant professor. What are the different aspects I have been working on? What are the different aspects of sugars I have been working on? What are the different functional aspects I have been working on? I will be explaining through you through my research topics. With this brief introduction, I just go into my talk. So these are the two important key players of my today's talk, which I will be interchangeably used throughout my whole lecture series. So I'll be talking about carbohydrates, which are otherwise called as glycans. So I will be briefly introducing about carbohydrates as well, and glycosidases and enzymes. So glycosidases are enzymes which acts on the carbohydrates, and they play a major important role. So for now, just have these two terms in your mind. So because these two terms has been going to be continuously will be taught by me throughout my lecture series. One is carbohydrates, which is otherwise called as glycans, and glycosidases. These are enzymes which acts on the carbohydrates. Let us see about carbohydrates. So carbohydrates, or otherwise called as a glycans, these are the abundant biomolecule on our planet Earth compared to other biomolecules which is known so far. So all must be knowing about different biomolecules in our planet. That includes nucleic acids, proteins, lipids, and carbohydrates. Of these four different biomolecules, carbohydrates are the abundant biomolecule in our planet. Another importance of a carbohydrates is that these carbohydrates are structurally diverse compared to other biomolecules which is available in our planet. So you can see, you can take a protein, you can take a nucleic acid, or you can take a lipids, and if you see the number of structures or possible structures which is available for which is possible for these biomolecules, you will be amazed to know that carbohydrates can display a variety of structures compared to other biomolecules, compared to what is known so far. So what determines the biodiversity of these polysaccharides? There are three important factors that determines the biodiversity. One is going to be the monosaccharide diversity. So that there are different types of monosaccharides which are being reported so far for the carbohydrates. That includes glucose, mannose, galactose. I can name them continuously. There are approximately 130 different monosaccharides which has been found in our planet Earth. So that itself gives a huge diversity for carbohydrates. The second level of diversity in carbohydrates is because of their connectivity. So the carbohydrates not only exist as a monomeric form or in a monomer form, these carbohydrates can be also connected or can be also linked to form a particular disaccharide, monosaccharide, or a polysaccharide via a specific glycosidic linkages. So seen over here is the two carbohydrates which is shown here, two disaccharide. These disaccharides are linked by these disaccharides composed of two galactose residue. As you can see that one galactose in this particular first compound, you can see that two galactose are linked by a 1,3 position, whereas this particular galactose is linked by a 1,4 position. So in a theoretically in a carbohydrate, you can have a linkage which can range between 3 to 6. You can have different types of linkages. Depending on the linkages, the carbohydrate functions are going to be changed as well. You see there is a subtle change in the structure, but their function is going to be changed. The third most important reason for structural diversity in the carbohydrates is their configuration. Carbohydrates can display two important stereochemistry, or they can import two stereo structural isomorphism. They are called as the alpha and the beta forms. So again, I'm taking the two examples over here, the two galactose residue, which are linked by 1,3 position. Again, the two galactose are again linked by a 1,3 position. And you can see over here is that now they have a different configuration. In one case, the carbohydrate is going to be in a beta form, and other case, the carbohydrate is going to be in the alpha form. And you can clearly see that again, slight structural change is going to give a diversity in the carbohydrate structure. Because of these three important parameters, approximately more than a millions of carbohydrate structures are possible in the nature. And because of the millions of carbohydrate structures, they are going to play very important or significant roles in biology. So this slide is basically a take-home message to say that the carbohydrates are the abundant biomolecule in our planet, and they display as a very vast sexual diversity compared to other biomolecules in our planet. Okay, so again, the importance of what is the importance of this structure? The importance of the structures of a carbohydrate is that a subtle change in the structure can create a huge impact in the biological process. As you might all know that, okay, plant cell is all covered by the plant cell wall. And if you dissect the plant cell wall, and if you see what is the major sugar which is present in the plant cell wall, it is all composed of cellulose. Cellulose, if you see in the monosaccharide form, this is all glucose. And when we combine this together, cellulose is linked via with the configuration of beta 1,4 glycosyl linkages. So in contrast, you see another polysaccharide over here, and this particular polysaccharide is starch. Again, the same polysaccharide is again still composed of a glucose, and again they are all combined in the form of 1,4 position, but not in the alpha form, but now in the beta form. There is a subtle change in the configuration. A subtle change in the configuration is going to create a huge difference in their function. In one case, when the glucose are connected via beta 1,4 linkages, this is going to have a very important crystalline property, which is going to be provide a very important structural support to a plant cell. Whereas when it is going to be connected in the alpha form, now it is going to be served as a energy source in this case, and this is also going to have a different mechanical and also elastic properties. So this is a very important slide, which is saying that a subtle change in the carbohydrate structure can have a huge impact in the biological process. Let us see now what are the different key functional roles of carbohydrates or glycans, which are known so far. So carbohydrates in the polysaccharide form or in the long chain of polysaccharide can provide structural support to a biological system. For example, plants, which I've already told you that plants, they are having two important biopolymers such as cellulose and chitin. These are all the plants has an important biomolecule called as the cellulose, whereas the fungal systems has an important biomolecule called as the chitin. They are all made up of a polymers. They are not a monosaccharide. They are all the polymers. They are going to provide a structural support to these biological systems, either in case of plant or in the fungal system. As a monosaccharides or oligosaccharides, which I have already told you, monosaccharides are the monomeric form of a carbohydrate. Oligosaccharides are the short chains of carbohydrates which are linked together. They do have important functions. For example, the monosaccharides and the oligosaccharides has been shown to be involved in energy or in the signaling processes. For example, you can take an important or a classic example for monosaccharide like glucose and disaccharide like sucrose or many other oligosaccharides. They have been important for energy sources. You might be well aware about the glucose and sucrose. Sucrose is a table sugar, and glucose is the monomeric form of a glucose, which provides instant energy when you are having. And some of the oligosaccharides or chitin oligosaccharides have been shown to involved in signaling processes. So these are the two important cases where you can see the carbohydrate functions itself. Either as a polysaccharide, they provide as a structural support, or either as a monosaccharides or a oligosaccharides, they are going to be involved in the energy or in the signaling process. So now the carbohydrates don't only associate or remain as carbohydrates, they can be also associated with the other biomolecule of interest. For example, carbohydrates can be also linked to a particular protein. Carbohydrates can be also linked to a particular lipid. So the example what I am showing over here is that a carbohydrate which is linked to a particular protein, which is going to dictate the protein functions. So some of the small oligosaccharides such as N-glycans and O-glycans are attached at a particular amino acid residue of a protein. For example, L-glycans are attached to the asparagine residue of a particular protein, whereas O-glycans are the short chains of sugars which will be attached to the serine threonine residues of a protein. And this is going to impart a huge important functions because they will be assisting a protein to get folded properly. They will be assisting a protein to get solubilized in a particular system correctly. They will also assist a protein to get folded properly. So these are the three important examples or the three important key functional roles of a carbohydrate, which are the main important functions of a carbohydrate, which is known so far. One is as the polysaccharides, they provide a structural support. As a monosaccharides, they are involved in energy. As a oligosaccharide form, they can be also associated with the proteins or other lipid forms. Okay, so these are the carbohydrates, and the carbohydrates, once they are synthesized in the system and they are present in the system, they are not always stable. These carbohydrates are acted upon by some enzymes which can cleave these carbohydrates during the life stage of any biological system. So the enzymes which acts on the carbohydrate are called as the glycosyl hydrolases or simply glycosidases. So these are carbohydrate active enzymes which cleave the glycosidic bond between the two carbohydrates or the two different sugars. I have already told you that two monomers are linked via a glycosidic bond. Glycosidases acts on the glycosidic bond and cuts them and release them or make them into a free sugar. So for example, take this case, for example, there is a biologically inactive compound. This is a salicylic acid, which is a very important hormone for plant defense. Salicylic acid is kept in an inactive form when a glucose is attached to it by a O-linked glycosidic bond. So glucosidases, or it's a glycosidase, the beta glucosidases can act on this particular glycosidic bond. It cleaves them and releases the salicylic acid. Now the salicylic acid can be free and they can perform their biological function. So this is one of the important role of the enzymes which acts on carbohydrates to make a biologically inactive compound to a biologically active compound. So another important role of glycosidases is plant cell wall remodeling. As I've already told you, plant cell walls have a lot of sugars. One of the important polymer which is present in plant cell wall are called as the xyloglucan, and there it has a lot of glycosyl linkages. These glycosidases acts on these sugar residues and they cleave the sugar residues, make the sugar residues free so that the plant can undergo a plant cell wall remodeling or the plant can adapt to a particular situations. So these are some of the few examples I have quoted, but there are multifaceted roles of glycosidases which acts on the carbohydrates which is present in the plant. And recently we have also reviewed on the different roles of glycosidases, and we have also published this review recently. I would also encourage the one who are seeing this video to read this publication, where we have put all the important multifaceted role of plant glycosyl hydrolases, especially in the context of plant pathogen interactions, and also covering a little bit of aspects of plant cell wall remodeling. So the basic message is that, okay, carbohydrates are present, and these carbohydrates are acted by a very important class of enzymes called as glycosidases, and these glycosidases play important role. Now the question is, do plant have only one glycosidases in them to break the carbohydrates for different functions, or plant has many glycosyl hydrolases in them? So for example, let's take a model plant. The model plant is the Arabidopsis thaliana. I am I hope everyone is aware about this particular model plant. It's a weed. So in the model plant, if you see the genome of Arabidopsis thaliana, in the genome of Arabidopsis thaliana, there are nearly 24 different glycosyl hydrolases families which are present. There are different types of glycosyl hydrolases family, and of the different glycosyl hydrolases family, there is only one family which is very well characterized, that is the glycosyl hydrolase 1. And majority of the other glycosyl families are not yet characterized. I have already told you about in the starting point that carbohydrate structures are highly diverse, and the diversity of carbohydrates reflects the number of glycosyl hydrolase genes which is present in the genome. So that is something very much amazing, right? Because carbohydrates are highly diverse in the plant, and this diversity has promoted evolution to have more number of glycosyl hydrolases genes in the genome of a plant, Arabidopsis thaliana. And this is not the case only with this plant, Arabidopsis thaliana. If you take a genome of a rice, or if you take a genome of any other crop plants, you will see different of glycosyl hydrolases from different GH families which are present in the plant system. But majority of these glycosyl hydrolases are not yet characterized or remains uncharacterized in the plants. So that gave a kind of a curiosity during my PhD studies, how to characterize them, because the broad range family, there are different types of glycosyl hydrolases are there, and different types of glycosyl hydrolases acts on different types of carbohydrates. So how to characterize them in a broadest way? So for this, during my PhD studies, we employed a technique which is called as activity-based protein profiling. So activity-based protein profiling, for the one who are new to this particular technology. So activity-based protein profiling involves usage of activity-based probes or activity-based chemical probes which acts or label protein in an activity-dependent manner. To say it very in a simple way, if I say, if you have an activity-based probe, and if you introduce an activity-based probe in a particular biological system, this probe will label only the proteins which are active, leaving behind the proteins which are not active. So this is something really, a very nice readout to see how many proteins are active in your system. For example, this is completely different from abundance-based technologies such as Western blotting, because in Western blotting, you will detect proteins. but you will not know if the proteins are going to be active. You can, of course, you can see that if in the Western blot, you can say that if the proteins are getting upregulated or downregulated, or they upregulated in activities or downregulated in activity, cannot be a readout of a Western blot. Whereas when you have an activity-based protein profiling, where you use an activity-based probe, the readout of activity-based probes will be that you will get a active proteins which can be monitored in a biological system. The main advantage of using the activity-based protein profiling technology is that activity of multiple proteins can be monitored in a native environment without the need for purifying the particular proteins. So activity-based protein profiling was exactly a very important technology, or very important technology for profiling glycosyl hydrolases, because I have already told you that glycosyl hydrolases are not one family, but there's a bunch of families in the plants. They are not yet characterized. There are multiple proteins in the plant. So applying activity-based protein profiling for profiling glycosyl hydrolases will be a great asset to characterize these important class of enzymes. So another important role of activity-based protein profiling is that they display the availability and also the reactivity of active site residues, which is a true important hallmark for a protein activity. So these are the two important advantages of activity-based protein profiling, and this is the hallmark for protein activity. So, so in my PhD, I was very much interested to profile plant glycosidases and applying activity-based protein profiling, which I have explained now. So, one fine evening, my supervisor came to me and he told, "Yeah, we have probes for activity-based protein profiling," and he gave me these three probes, and these three probes have been synthesized by the chemist, and these probes are potentially is going to target different glycosyl hydrolases. I was very much interested. I took this probe and I tested if at all this probe is going to act on glycosyl hydrolases or not. So before going, what happened when I used this probe to label the particular plant proteome? Just briefly, I would like to say how the probe, what is the structural elements of the probe. The probe consists of three important structural elements. For the first is going to be the warhead. So warhead is a region which is going to go inside the active site pocket of a particular enzyme. So in this case, for profiling glycosidases, chemist has put a warhead with the cyclophenol aziridine group as a warhead. Cyclophenol aziridine is a group. It's a kind of a sugar-like compound. It's a sugar-like compound which is a powerful inhibitor of glycosidases, which can easily go inside the active site of a particular enzyme. So now, if you have this probe, if you spike this probe into a biological system, the probe will take the probe will go inside the active site of a glycosidases and it will be labeled. And the probe has a second element which is called as the linker. In this case, the linker is going to be the triazole linker. The linker is going to link a particular reporter tag. So JJB70 is a probe which has a reported tag of BodyPy. So the proteins can be easily visualized by fluorescent scanning. So BodyPy is a fluorescent tag. So whenever the protein is going to be labeled, you can do a in-gel fluorescent scanning and you can see what are the proteins which are being labeled with the help of a BodyPy tag. And biotin is another tag. So when the proteins have labeled a particular active glycosidases, now with the help of biotin, you can pull down the protein and you can identify what are those glycosidases with the help of the mass spectrometric analysis. The another probe is called as the KY371 probe, and this probe does not have any reported tag like biotin or BodyPy, but it has an alkyne tag, and these probes can be useful for click chemistry and it can be used in the native system. So if you have this probe, if you spike it in a native system, the probe will label a protein, and later on you can tag or click the reported tag in the later on in the following stages. So the image which is shown in the right hand side is the scheme, how a probe labels a particular glycosidases. If you have a probe and if you treat it in a biological system, the probe will go inside the catalytic site of a particular enzyme. In this case, it's going to be the glycosidases, and the glycosidases has a nucleophilic residue with a glutamate residue. The oxygen of a glutamate residue will form a nucleophilic attack at the aziridine ring. As a result, you will be having a covalent bond formed between the probe and the protein. And the probe is having a tag. It could be a BodyPy tag or a biotin tag or alkyne tag. Later on, if you have a BodyPy tag, by using fluorescence, you can monitor the fluorescence. If the probe is having a biotin tag, you can pull down and identify the particular proteins. So this is the basics about the activity-based probes for glycosidases and the mechanisms how our activity-based probes works to profile glycosyl hydrolases. So now I have got the probe from my PhD supervisor, and now what I did to test, I used Arabidopsis thaliana as a model system. I took the Arabidopsis thaliana because I have already shown you that in Arabidopsis thaliana, in the genome, there is a lot of glycosyl hydrolases. We were very curious to know what are the glycosylases which are active, or what are the glycosylases which can be labeled with the help of the JJB70 probe. I took the Arabidopsis thaliana leaf extract, grinded them in a suitable buffer, and added the JJB70 probe and profiled them with the help of the in-gel fluorescence scanning, which I have applied, which I have told you now. So compared to the no probe control, we were able to see bunch of different glycosyl hydrolases, which was labeled with the help of the JJB70 probe, and these labeling of these signals could be suppressed with the help of the inhibitor. So this was clearly saying that, okay, the proteins which are labeled by JJB70 or glycosyl hydrolases, and now we got curiosity to know what kind of proteins are labeled with the JJB70 probes. I have already told you to know the proteins which are labeled by the activity-based probes, you need a biotin tag. So we took another probe, which is JJB11 probe, which has the biotin tag. Again, we took the proteome labeled with JJB11, and then we did a pull down with the JJB11 protein, and we did a normal SDS page, and with the help of CyberRuby, we are able to see different type of glycosyl, different type of proteins which are enriched over here. And then we cut out these bands and we did a mass spectrometry analysis. We were amazed to say that many of the proteins, or majority of the proteins, were glycosyl hydrolases. To show you the brief classification, here we have identified 20 different glycosylases belonging to different families. To show you what are the different families which we were able to target with the JJB70 probes, we were able to target proteins which is belonging to the family 1, 3, 35, and 79. And also these family members were targeting different glycosid substrates. For example, we could see an enzyme which is targeting a glucosidase with targeting a glucoside. We could see an enzyme which is targeting a xyloside as a xylosidase. We could see a galactosidase and a glucuronidase in our data set, which is indicating that the JJB probes, that the activity-based probes, target the glycosyl hydrolases. Also, we went in detail about the mass spectrometric analysis, and we could see which is the site of action, or where this probe is exactly labeled. With the help of mass spectrometry, we were able to see the B ions and Y ions. So B ions and Y ions are, by calculating the masses of BS and Y ions, you can really predict what kind of residues are labeled with the probe. We were able to clearly see that it is labeling the glutamate residue, which is on the active site of a particular protein. And based on this, we have validated our system that JJB probes, or the activity-based probes, target glycosidases, and it acts as the mechanism which is predicted to label the glycosidases exactly in their active site point. Okay, so having validated these glycosidases, next I applied these, during my PhD, I applied these probes to profile an important compartment in a plant, which is called as the apoplast. So as you might all know that apoplast, when you take a plant leaf and you make a cross section, you will see different types of cell types. You will see epidermal cells, you will see a mesophyll cells, but these cells are not all clumped together. They're not all joined together, but there is a space between these cells, and the space between these cells together with the cell wall, they constitute the apoplastic region. It's a very important component in the context of plant pathogen interaction, because the initial communication between a plant and the pathogen is initiated in the apoplast. And also this is an important compartment which is heavily colonized by microbes during the infection cycle, because this is the apoplast where there is a nutrient transport and also water transport which is going on. These pathogens will take these nutrients and they will rapidly multiply during their infection cycle. So, and also this is not a very easy compartment to survive for the pathogens which are colonizing these apoplast. The pathogens could be a bacterial pathogen or it could be a fungal pathogen. They are going to face a hostile environment in the apoplast. The hostile environment in the apoplast is all majorly because of the hydrolases the plant is going to secrete in this particular compartment. For instance, let's say a plant leaf is colonized by a fungal pathogen or a bacterial pathogen. The plant will make the environment more hydrolytic by secreting a lot of hydrolases into the compartment, by secreting glycosyl hydrolases. The plant will have more glycosyl hydrolases in the particular environment. So now next step question, what I was interested during my PhD was, what is the role of these glycosidases what is the role of these glycosidases during the bacterial pathogen interactions. So as a bacterial pathogen model system, we use Pseudomonas syringae as a model system. Pseudomonas syringae DC3000 causes a classical bacterial speck disease in tomato and also in many other Solanaceae crops, and also it causes a lot of it's an economically important plant bacterial plant pathogen. We use this particular plant pathogen as a model system to see what is the role of glycosidases. So during the infection of a Pseudomonas syringae, and we use Nicotiana bentamiana as a model system to study. I've already told you Pseudomonas Nicotiana bentamiana can be infected by Pseudomonas syringae pathowar, and it causes a classical bacterial speck diseases. To study what is the role of glycosidases during plant bacterial interactions, what I have already told you, we use Nicotiana bentamiana as a model system. The main reason why we chose Nicotiana bentamiana is that the leaf size is very big, and it is also a very interesting model system to study apoplast in a plant, because you can get sufficient proteomes from the apoplast with less cytoplasmic contamination from this Nicotiana bentamiana, because the protocol for isolation of apoplastic fluids is very well established, and you get majority of the proteins which you are getting from the Nicotiana bentamiana are going to be apoplastic proteins. So in order to know what are all the glycosidases, or what is the status of glycosidases during the plant pathogen or during the bacterial interaction, what I did was I infiltrated the Pseudomonas syringae strains. The first strain is going to be PTO DC3000, which is a wild type strain which causes cell death when injected into Nicotiana bentamiana, and the other strain is PTO DC3000, which is Delta Hopq strain, which causes bacterial disease when infiltrated into the bacteria. And these two particular strains were infiltrated into the Nicotiana bentamiana leaves. And after 2 DPI, I isolated the apoplastic fluids, which is enriched with the glycosidases, and labeled with the JJB probes, which targets the glycosidases, and ran a normal USDS page and did an fluorescence scanning to see what is happening during the bacterial infection, what is the difference between before infection and after infection. So this is the gel which I obtained after the collecting the apoplastic fluids from the infected and also the non-infected plants. So these are the first these are the two samples. This is the untreated and the mock treated plants. This is just infected with the water or infiltrated with the water. You can see this is the general profile of glycosidases which was seen. Interestingly, the profile upon infection with the wild type of the Hopq strain, we were able to see that several bands were weakly labeled during infection of this particular pathogen, and also we were able to see distinct signals which was appearing as a result of infection with the wild type strain. So during my PhD, I focused on this particular protein, and this particular protein, we tried to identify what is this particular protein, and this particular protein was I have already told you how we have identified. Again, I'm briefly saying we used a JJB 111 probe, we did a biotin pull down, we did a mass spectrometry, and we identified what is this particular protein. This protein corresponds to a particular enzyme, which is called as the beta-galactosidase enzyme from a family GH35. So you see that this particular protein's activity was going down upon infection, and maybe that is the reason it has a reduced labeling. So the reduced labeling could be also due to another reason, that their abundance, or they might be down-regulated during the infection. Because of their down-regulated in abundance, their labeling is also weak. That could be also another reason. So to know if at all the abundance is the major reason why a beta-galactosidase is weakly labeled or reduced labeling during infection, we did a proteomics on the infected versus the non-infected plants. We were amazed to see that that the beta-galactosidase enzyme, which are present over here, their abundance was not changed during infection at all. The abundance remained the same, but their activities was going down during infection. So that gave a very nice, interesting turn, that made me a curiosity to go further into this particular protein to investigate if there is any inhibitor in the sample. Because of the inhibitor, is this the inhibitor? Because of the inhibitor, this particular protein is getting inhibited, or the activity of this particular proteins are reduced. So to do that, I standardized an approach which is called as the convolution ABBP. The approach is very simple. So what you do, you take two different proteomes, you mix them, and you label with a JJB70 probe. And as a second sample, what you do, you take the two proteomes, you label with the JJB probes, and then you mix them and you run it on a gel. So this is the approach. This is how it looks like. This is the profile of a non-infected plant, and this is the profile of an infected plant. Upon infection, you see a reduced labeling. So now what you do, you collect the proteomes, you collect the proteomes together, and you mix them and then you label, or you label the proteomes and then you mix. So when you label the proteomes and then when you mix, if at all you cannot see the inhibitory activity, as a result, you see that the intensity is not drastically reduced. Whereas when you first mix the proteome, and if at all there is an inhibitor present inside the inhibitor, if at all there is an inhibitor present inside the infected sample, this will cause suppression of signal over here. And this suppression of signal indicates that there is a inhibitor present inside the sample of interest. So, so exactly we got the same results, that there was an inhibition when we mix and label our sample. This is indicating that there is an inhibitor which is present in the infected sample, and this particular inhibitor is inhibiting the beta galactosidase enzyme. Okay, so why does the beta galactosidase is getting inhibited during a plant pathogen interaction? If at all this particular enzyme is targeted by a pathogen, or this particular enzyme is inhibited during an interaction, this particular enzyme should be an important enzyme or important immune enzyme. To test if at all this particular protein is an important immune enzyme, we used a gene editing approach. We gene edited, or we did a knockout using a CRISPR-Cas9. We generated two important lines. We generated BGAL1 and BGAL2 line, which had an insertion, which has a deletion mutation such that the protein is going to be truncated. And then we collected the apoplastic fluids, and we labeled the apoplastic fluids of a wild type and also the mutant plants. We were able to see that the band signal, that the activities which is displayed by beta galactosidase is gone, because the knockout is perfect. And we used these two lines and we did a pathogen infection assay. Upon doing a pathogen infection assay, we were amazed to see that the pathogen was growing more. It was growing a log-fold more on the plants which is devoid of beta galactosidase enzyme. So, or in other words, you can say BGAL mutants are less resistant to Pseudomonas syringae, or BGAL enzyme it's an important immune enzyme which is important for resistance against the bacterial pathogen. So how does this BGAL actually contribute to immunity? What is the role of a beta galactosidase enzyme? For this, we devised a very interesting approach. What we did, we overexpressed the beta galactosidase enzyme in the apoplastic fluids by using an agroinfiltration approach. We took the apoplastic fluids, which is either containing the beta galactosidase enzyme or no beta galactosidase enzyme. We treated with the bacterial pathogen, that is the Pseudomonas syringae. We incubated this for some time. And after this, we collected the bacterial pellet and spiked it onto a leaf disc, which is having two important substrates, that is luminol and horseradish peroxidase. And this is a very important classical plant immunity assay to test if at all there is an elicitor which is released upon action of any beta galactosidase enzyme, or if at all there is a elicitor which is present in your biological sample. So for example, if at all there is a elicitor which is present in the biological sample, this elicitor will bind to the PRRs, that is pathogen recognizing receptors, which is present on a leaf disc, and this particular receptor will receive the elicitor, and after receiving the elicitor, this will trigger the PTI responses. Together with that, an important enzyme will be activated, that is called as the RBOHD. This will activate the reactive oxygen species production, and this reactive oxygen species production will illuminate the luminol, or it will activate a luminol. As a result, you can monitor the classical luminance which is going to be generated. If there is a luminance which is being generated, this is an indication that there is an elicitor which is present in your sample of interest. So having this pipeline in the mind, so we went inside and then we looked what is happening during the bacterial and the BGAL interactions. We found that when you take a normal wild type bacteria and when we put it on the leaf disc, this is not being recognized, so there is no any rosbush. Whereas when you treat the bacteria with the beta galactosidase enzyme and then you take the treated bacteria and put it on the leaf disc, we were able to see a burst which was coming, which is indicating that there is a elicitor which is released from the bacterial pathogen. Whereas now if we add an inhibitor of the beta galactosidase, which is going to be the galactostatin inhibitor, which is going to be very specific to inhibit the beta galactosidase activity, we can see there is no any response. So this is a very indicator, and this is a very starting point to clearly clarify that the beta galactosidase enzyme, which was suppressed during the infection, is actually targeting the bacteria and it is releasing a particular inhibitor. And with this data, we don't know what this particular inhibitor is. So we tried the same thing in Arabidopsis thaliana, and in Arabidopsis thaliana, again, we got the same results. You see that whenever the bacteria is treated with the BGAL enzyme and spiked it on the Arabidopsis thaliana leaf, you can see a huge burst which is coming out. When you don't treat it with the beta galactosidase enzyme, you don't see a particular burst. So now having this particular, the advantage of having an Arabidopsis thaliana is that there are different mutants of Arabidopsis thaliana which are available. And now you can take the treated bacteria and be tested against different mutants. So I'm not showing all the mutant collections what we all screen, but one of the mutant did not respond even though if you treat it with the beta-galactosidase enzyme, and that particular mutant is the FLS2C mutant. So FLS2 is a gene, and this is a receptor for flagellin, is a flagellin receptor. So whenever the plant is devoid of a flagellin receptor, it is not getting recognized, or it is not recognizing the elicitor, which is released by beta-galactosidase enzyme. So this was giving an indication that beta-galactosidase is somehow related with the flagellin protein. And now when we took the bacteria, which then we took an advantage of bacteria which has the flagellin or which don't have a flagellin. So delta HQ is a bacteria which has a normal flagellin, and delta HQ free C is a bacterial mutant which does not have a flagellin in them. So now when you take a normal bacteria which has a flagellin, and when you treat it with the beta-galactosidase enzyme, you see a huge burst which was coming from the Arabidopsis leaf. Now if you take a bacteria which is a mutant which does not have a flagellin protein, and when you treat it with the bacteria with the B-gall enzyme, we could not see the bacterial burst which was coming. This is the same case with the Nicotiana bentamiana. This is clearly indicating that beta-galactosidase is acting on a flagellin protein. So now that is an important thing. So beta-galactosidase is an enzyme which acts on sugar, but flagella is a protein. It's made up of flagellin proteins. How these two are getting connected together? So we can go in detail and see how is the flagellin protein actually composed of. So flagellin proteins are composed of flagellin peptides, but these proteins are actually buried inside. But on the top, you will see a lot of sugar residues which is exposed in the flagellin proteins. And these particular sugar residues are nothing but the O-glycans. So I already have introduced about a topic about O-glycans when I started this topic. So O-glycans are small oligosaccharides, a short chain of sugars which is attached to a particular residue of a particular protein. So in this case, the O-glycan is going to be with the three important sugars, that is 2 raminoses and 1 biosamine is a 3 important sugar, and this is attached to a serine residue over here, and this is making a O-glycosidic linkage, and this is a O-glycans which is present in the flagella. So now we thought, okay, this could be the this could be the substrate of a beta-galactosidase enzyme, that the beta-galactosidase enzyme is acting on this particular glycosidic linkage somewhere, and that might be the reason. And we took now an advantage of a bacteria which is called as Pseudomonas syringae pathowort tabaci, that is 6605, where we could get the mutants of the flagellin glycosylation mutants. So here you can see that this is a wild-type bacteria, and we take the wild-type bacteria, and when you don't treat it with enzyme, you see a very small response over here. And so this is also very interesting because this particular wild-type bacteria does not produce an inhibitor. When it is not producing an inhibitor, and when you have apoplastic fluids which is not having the overexpressed B-gall but the endogenous B-gall, still we were able to see a small burst which is coming because the bacteria is not producing an inhibitor. As a result, the endogenous beta-galactosidase enzyme has processed the particular has released the particular elicitor. Thereby we are seeing a small burst. Whereas when we overexpress the beta-galactosidase enzyme, upon overexpression of a beta-galactosidase enzyme, this has resulted in the production of huge elicitor which is released from the bacterial flagellin, and this we were able to see as a huge burst over here. This is a case with the wild-type tabaci strain. Now we took an advantage of a mutants which are going to have mutants in the enzymes which are it's a biosynthetic enzymes which is not making these particular glycans. So when we have the FGT1 mutant where you don't have the O-glycans at all, either you treat it with bacteria or you don't treat it with the bacteria, the apoplastic fluids are able to process the flagellin, and it is going to get recognized in the leaf. As a result, there is no difference in their perception. You see the same response is going to happen. Now, when you have the mutant of VIOT, that is the VIOT transferase, which adds the lost residue, the M biosamine. If you have a mutant over here, and when you again treat it with, or we don't treat it with bacteria, the plant is able to perceive this particular glycan already. So this is indicating that Y biosamine is a very important residue over here, and this has to be cleaved by the enzyme, which is going to be the beta-galactosidase enzyme. Interestingly, we tried another bacteria, which is a Pseudomonas syringae pathowort syringae, B72-8A. It's an important Pseudomonas syringae strain, which does not have VEM biosamine, but it has a putative sugar, which is called as the glucan. This is N-acetyl glucosamine sugar as a terminal sugar over here in the O-glycan. So now when it has a terminal sugar of N-acetyl glucosamine, either you treat it with the B-gol enzyme or you don't treat it with the B-gol enzyme. And if you put it on in the Arabidopsis leaf, you don't see any burst at all. So this is a very interesting thing, that this particular bacteria has evolved and it has now put an interesting isoform or an interesting glycoform in their glycan, that just changing one sugar residue, this is not going to be able to be perceived by the plant immune system. And this is an important strategy which a bacteria follows. So in this slide, you can already see importance of a sugar in the bacterial perception, or importance of a sugar during plant pathogen interaction. Just change in one sugar residue, there is a change from M-viasamine to a glucnac, the plant is not able to be recognizing this particular pathogen, leading to more susceptibility. And whereas when you have a M-viasamine, this M-viasamine can be treated, can be cleaved by B-gol enzyme, leading to the huge production of the elicitor, which is released from the bacterial flagellum. Okay, to conclude this particular story, I have already told you the normal bacteria, it has this kind of O glycans. It has a two raminose, which is attached to a serine residue, followed by a terminal M-viasamine, which is going to be over here. Whenever the bacteria is exposing this particular flagellin, plant secretes beta-galactosidase enzyme, and beta-galactosidase enzyme is going to cleave the M-viasamine sugar. It is going to release the M-viasamine sugar. Once the M-viasamine sugar is released, and now the bacteria is going to have only raminoses and flagellin protein, and this raminoses and the flagellin will be cleaved by other glycosyl hydrolases and proteases. This is going to lead to a release of flagellin peptides, and these flagellin peptides are received by FLS2 receptor. To circumvent this, bacteria is using sugar as an important shield to prevent this particular recognition process. For example, let's say in the normal case, if this particular catalytic events are happening, the plant is going to be resistant, because the plant upon recognizing the flagellin peptides, the plant is going to be resistant because it is recognizing the flagellin peptides. Now what the bacteria will do, the bacteria will try to make a strategy in a way that it's not going to get recognized. And in our paper and in our work, we have shown two important strategies. One strategy is to have an important glycoform. Now a bacteria, instead of having M-viasamine, which is the target of beta-galactosidase, now the bacteria is going to have another sugar, which is going to be the glucnac sugar. By having another glycoform, the plant is not going to get recognizing this particular pathogen, and the pathogen is going to grow very fast, leading to susceptibility. And second important strategy is to use a beta-galactosidase inhibitor. I hope you remember that upon infection, by convolution ABPP, we have shown that there is an inhibitor produced in the sample. So now I'm not going to discuss in detail about the other approaches, but we have further validated and seen that the bacteria is actually producing the beta-galactosidase inhibitor. By inhibiting the beta-galactosidase enzyme, by using the B-gol inhibitor, the bacteria is preventing the release of flagellin peptides. So recently we have also, by using the forward genetic approach and also biochemical purification, hardcore biochemical purification, we have purified this particular inhibitor, which is produced by the bacterial pathogen, which is Pseudomonas syringae, and the bacterial, then the sugar is going to be, we have named the sugar as a glycosyl protein, which is an immunosugar. So this is not a sugar exactly, and this is called as a immunosugar, because instead of oxygen, which is present in the in the 5-member ring, it is going to have a nitrogen over here. So now you can just see that how clever the bacteria is. Now the bacteria is not making the substrate, but the bacteria is making a substrate mimic, which is going to be an inhibitor of a beta-galactosidase enzyme. As a result, the beta-galactosidase, which is an important immune enzyme, is going to get inhibited. As a result, the plant is going to be more susceptible. So this is the part which I have been carried out during my PhD, and during this you can see the importance of different sugars, especially M-viasamine, glucnac, and also the beta-galactosidase inhibitor, how they are all coming in the context of plant-bacterial interactions. So let us shortly see what happens in the apoplast when the fungi is going to get colonized. I have already told you that the apoplast is not only the site for bacterial multiplication. Apoplast is also a site for fungal multiplication. So when the fungi is going to go inside the apoplast, the fungal extracellular polysaccharides are the first important glycans or the first important carbohydrates which is going to get exposed in the apoplast. They are going to get exposed, or going to get the first initial contact, or going to get touched in the apoplastic region. So whenever I say fungi and whenever I say fungal extracellular polysaccharide, the most important thing which comes to everyone's mind is the chitin. Chitin is a usual suspect in the fungal cell walls, and the chitin, which is a polymer of N-acetyl glucosamine units, but this is not true. Chitin is a very minimal polymer, and this is the chitin is going to be present in a very minimal amount in the fungal cell wall, because the recent structural data has indicated that chitin is not the abundant sugar which is present in the fungal cell wall, but the beta glucans are the one which is present in the fungal cell wall. Chitin is actually sandwiched inside the inner core of the fungal cell wall membrane, but the majority of the space which is occupied by the fungals in the fungal cell wall are the beta glucans. That is going to be the beta 1,6, 1,4, and 1,3 glucans, or the major neutral sugar glycans which are filling the space in the fungal cell wall. Or in the other words, you can clearly see that chitin is not the important cell wall component of the fungal, but the beta glucans are the one which is the important component of the fungi. And the majority of the beta glucans or the functions of the beta glucans are not yet studied in the context of fungal cell wall biology. So during my postdoc in Professor Alga Sukara's lab, we took this challenge to characterize what is the role of beta glucans in what is the role of beta glucans in fungal cell wall. For this, we took an important fungi which is called a Serendipita indica. Serendipita indica is a very important endophytic fungi, and this Serendipita indica, whenever it is being treated with the plant, it enhances the plant growth. It's a plant growth promoting bacteria fungi, and this particular fungi colonizes the roots of several plants, and it increases the biomass of the particular plant, both at the root level and also at the shoot level. So and also apart from increasing the root biomass, and the Serendipita indica is also a very important fungi, because it can also provide local resistance. So in this case, it has been previously shown that the Serendipita indica can also prevent the infection of Rhizoctonia root rot, and also it can also provide infection against biotrophic pathogen Bloomeria as well. So we took this particular fungi, and we used two important dyes. So one dye is called as the WGA dye, which binds to chitin, and the other dye is FGB1, and this is a lectin which binds to the beta glucans. And we performed a confocal microscopy. We were amazed to see that we can monitor the two important polysaccharides in our samples. So the red one, which is labeled over here, this is the chitin layer, which is made up of N-acetyl glucosamine layer, and we were able to see that the FGB1, and that is the lectin, which is labeled in the green fluorescent dye. You can see there's a copious amount of polymers of which is made up of glucans are actually encapsulating the chitin layer. And this is all the extracellular polysaccharides which are produced by the fungus. The extracellular polysaccharides are very much known in the plant bacterial field, but in the fungal field, the fungal producing the extracellular polysaccharide matrix is something new, and this has not been explored in detail so far. So what we did, we collected the extracellular polysaccharides from the fungi, that is going to be the Serendipita indica, and we performed a glycomic analysis. So these are the two important glycomic approach what we are following in the lab, and also in our current lab we have already established these techniques. So first we followed the monosaccharide composition. The monosaccharide composition, you take the glycans, you hydrolyze the glycans with a mild hydrolysis with the TFA, and you do reduction and acetylation, and you convert a sugar into a alditol acetate, and you perform GC-MS analysis. And in this case, you will get what are the sugars or what are the monosaccharides which is present in the EPS matrix. And the second approach what we followed was the linkage analysis. In this case, you methylate a particular glycan. After methylating, you do a reduction and also acetylation. You convert a particular glycan into a species called as partially methylated alditol acetates, and this partially methylated alditol acetates was spiked into GC-MS, and we have done a GC-MS analysis, and we have made up a library of different sugars and also different sugar linkages. And with these two important approaches, you will know what are all the monosaccharides which are present in the samples, and also what is the linkage of these particular sugars are. For example, let's say this is a small oligosaccharide over here, and if at all you wanted to know what is the linkage which is present over here, and this particular analysis can say this is a particular linkage. So if at all I am taking this oligosaccharide and performing the linkage analysis, this analysis will say me this particular oligosaccharide has a 3-linked glucose, because you see here this is a C1 carbon and this is linked to a third carbon. This will say this is the 3-linked glucose. By having this particular idea, now you can postulate an hypothesis. If it is a 3-linked glucose, it has to be a beta 1,3 glucan. So this can allow you to make a hypothesis to know what are the polysaccharides which are present in your sample of interest. So having this particular approach, we took a cell wall of a Serendipita indica and also took the cell wall EPS from the Serendipita indica, performed the glycome analysis. We were able to see that their composition was distinct. The cell wall, we got a very high concentration of 4-link glucoses, whereas in the EPS matrix, we got a very high concentration of 3-link glucose and a 3,6-link glucose. So the 3 and 3,6-link glucose are the biomarker glycosidic sugars for beta 1,3 and 1,6 glycans, and which is indicating that EPS matrix has the different composition than cell wall, and EPS matrix has a 3 and a 3,6 glucose at a beta 1,3 and beta 1,6 glucans are very much high in the EPS matrix of Serendipita indica. So whenever a polysaccharide is present, and when this polysaccharide is present this polysaccharide is represented in the apoplastic region, plant secretes, I have already informed you, they produce a lot of enzymes, especially the glycosyl hydrolases. So what is known so far in the plant fungal interaction field is that plant secretes a lot of enzymes which is called as the chitinases, and these chitinases acts on chitin, and they release a chitin oligosaccharide, in this case the hexamers of chitin, that's a chitohexose, and these chitohexose are recognized by the PRRs, that's the pathogen recognizing receptors, and that leads to activation of enzymes, which is the RBOHD, and this will lead to a formation of a reactive oxygen species, and this will be further converted into hydrogen peroxide that will activate several receptors in the plant membrane, and ultimately it will have a robust immune responses. You see that chitinase is going to release a fragment upon recognition, is going to have a strong immune responses. But now we have already shown that apart from cell wall, plant fungi is also having an extra layer on the top, and this extra layer is called as the extracellular polysaccharide matrix, and this is made up of beta 1,3, 1,6 glucans. So which enzymes are going to target this particular glucans? We were interested to know. For this, what we did, we performed a quantitative proteome analysis of apoplastic fluids. We took a barley plant and we treated with the Serendipita indica. We treated with the Serendipita indica, that's the fungal spores, and we collected the apoplastic fluids at 3 different time points. We collected the apoplastic fluids at the 3-day, 10th day, and the 14th day. That's the 3 different time points. And after collecting the apoplastic fluids, we performed the label-free quantitative proteomics. So performing a label-free quantitative proteomics, we were able to see different K-zymes or different glycosyl hydrolases which were upregulated in the apoplastic fluids of barley. So in the 10th DPI, that's a 10-day post-infection, and the 14th day post-infection, we were able to see one enzyme which was represented in the 2 important time points. And this enzyme is the beta 1,3 endoglucanase, represented as BGLU2, and again the beta N3 represented as a BJ1. It's the same enzyme which was getting highly accumulated in the apoplastic fluids at the early and also in the late stage of infection. So we purified this particular enzyme and we treated with the cell wall and also the EPS fractions. We analyzed what kind of fragments are released by the action of hydrolytic action of this particular enzyme. We were amazed to see that in the EPS matrix, that's the extracellular polysaccharide matrix, HVBGLU2 was releasing a fragment, a high molecular weight fragment very clearly in a very high intensity from the EPS matrix, whereas this particular matrix was absent when this enzyme was treated with the cell wall. So this is indicating that this is an enzyme which is very specific for EPS matrix, and this particular enzyme is releasing a high molecular weight fragment very specifically for the extracellular polysaccharide matrix, but not for the plant cell wall. Now, when you take a commercially available enzyme, that is 2 important enzyme, 2 interesting enzyme, which is trichoderma lysing enzyme and helix spermatia enzyme. These are all random cutters. These are not biologically relevant enzymes, but they cut beta 1,3 glucans. When you take this enzyme and when you put it in the EPS matrix, you see that there are a lot of fragments which is coming because they are not biologically relevant, but they are randomly cutting the glucans which is present in the cell wall. They release a lot of fragments over here. Whereas when you treat it with the HVBGLE2, they released only a very specific fragment from the EPS matrix of Serendipita indica. So that gave a curiosity to know what is this particular fragment. Again, I'm not showing how we purified this fragment. We used a lot of glycan analytics and chromatographic techniques to purify the fragment which is released from the extracellular polysaccharide matrix of EPS, and then we treated with the barley roots. I've already told you this particular experimental setup for the leaf. The same thing we established for the barley roots, and we treated with the barley roots to see if at all the fragment is going to trigger immune responses in the barley root, or is going to trigger a pattern PTA responses in the barley root. So what we did, we tried taking the barley root and we tried it at 2 different concentrations at 30 micromolar and 300 micromolar. The result was no. We did not see any burst at all. The fragment was not getting recognized by the fragment at all. So we were about to give up this project, okay, because we thought, okay, this is going to be an important fragment, and this particular fragment is going to recognized by the particular receptor in the roots. Thereby, it's going to trigger an immune system. But we couldn't see any burst at all. We couldn't see any immune responses in the barley roots at all. Whereas when we treated the barley roots with the chytohexose, we saw a huge burst, which is still saying that the barley roots is able to perceive the elicitor, but not the fragment which is released from the EPS matrix. So what we did, okay, we tried like combining the chytohexose, which is an elicitor, with our fragment. We thought, okay, when we combine them together, we are going to get a more immune responses. Whereas we got it other way around. We saw that when we combine the fragment with the chytohexoses, the elicitation, the ROS which is produced by the chytohexose was going down in a concentration dependent manner. You can see that the chytohexose with 30 micromolar showed a very few bursts. When it is at the 300 micromolar, which is a very high concentration of the fragment, there is no burst seen at all. And this is indicating that the fragment which is released from the EPS matrix has a ROS scavenging activity, or is able to scavenge the ROS which is produced in the apoplastic space. And this is a very clever strategy which the fungi use, that it is hijacking a enzyme which is present in the plant to degrade its own EPS matrix to release a susceptible factor which can scavenge the ROS such that the fungi can survive in the apoplast. Because I have already told you that apoplast is a hostile environment which has a lot of hydrolytic enzyme and also the reactive oxygen species. Now the fungi can easily survive in the apoplast by using by hijacking the HVBG2 enzyme. So, and also like we took the fragment and also we treated with the Serendipita indica roots, and we performed a colonization assay. We were able to see that the fungi was colonizing more when you have more fragment. So when the fungi is able to when the poly roots are treated with this BGD with the two different concentration, we can able to see that the fungal colonization was high. And this is indicating that this is the ROS scavenging factor which is released by the released from the EPS matrix is the susceptibility factor. So we used a lot of analytics to know what is the structure of this particular compound. We used a MALDI-TOF analysis, and we found that this structure has 10 different sugars. So how are these particular 10 different sugars arranged? We use the NMR spectroscopy to know what kind of sugars are there. We were able to see a very clear spectrums which is corresponding to beta-1,3 and beta-1,6 glucans. And based upon the NMR and the MALDI data, we already came up with the partial structure that the sugar is going to have 7 beta-1,3 linked glucans substituted with singly substituted with beta-1,6 glucosyl linkages. To confirm our NMR data, we also use two important enzymes. One is GH17A and GH30, FBGH30. GH17A will not cleave particular glycosidic bond whenever there is any substitution on a particular sugar. So when we take this particular fragment, when we treat it with the BH17A, we couldn't see any mass shift, which is indicating that this particular beta-1,3 glucan is substituted one. And when we now treat it with the GH70 enzyme, GH30 enzyme, this is an enzyme which is very specific for beta-1,6 glucosyl linkages. We were able to see a shift of 3 sugars. The sugars was the Daltons was a shift of 3 important sugars. This gave to the rise of a hexose form. So this is indicating that there are 3 sugars which is sitting on the top of the particular beta-1,3 glucan. When we mix these 2 glucans, we were able to see that the whole fragment is completely digested. This is because GH30 will remove the beta-1,6 linkages and the linear beta-1,3 will be cleaved by GH17A. The fragment is gone. So based on this, we have already confirmedly say that this is the fragment which is released from the fungal EPS matrix, and this particular fragment is having a ROS scavenging activity. So why does this particular fragment has the ROS scavenging activity? We take this particular fragment and do a Fenton reaction which produces the reactive oxygen species, and you can see that this particular fragment is highly reactive to the Fenton. As a result, you will not see any fragment because it's already degraded. It's a reactive with the reactive oxygen species. Whereas now you prevent this particular reaction by adding EDTA, the fragment is coming back. So again, this is another way to say that this has an antioxidant property. And to address what is the structural element, what is the important structural element which is important for the ROS scavenging activity, we took a normal glucan which has a 7 beta-1,3 linked glycosyl linkage. You see that 1, 2, 3, 4, 5, 6, 7 is only with beta-1,3 linkages. And when we performed a Fenton reaction, we could not see any degradation of this particular fragment at all. So this is indicating that the beta-1,6 linkage which is on the top of the beta-1,3 is an important factor by which the pathogen employs to scavenge the ROS which is present inside the apoplastic space. Okay, so this is a summary of the model what we have found. So usually it is already known that glycosyl hydrolases like chitinases acts on chitin to release the chitin PAMs recognition to activate the immunity. Now the fungi, that the beneficial fungi is having an additional layer which is called as the extracellular EPS matrix, and the fungi is also hijacking a protein which is the HBBGL2 enzyme to degrade its EPS matrix to release the ROS scavenging factor, and it is going to scavenge the ROS. As a result of scavenging of a ROS will not be converted into hydrogen peroxide, and these immune receptors will not be activated, leading to weak immunity or enhanced colonization. So you can just see how important sugars are playing a role during the plant fungal interactions, especially during the beneficial fungi interactions. So now I have told you about the beneficial fungi. What about the fungal pathogens? Do fungal pathogens make extracellular polysaccharide matrix, or what is the enzyme which is targeting the fungal extracellular matrix? Do fungal pathogens also follow the same strategy to scavenge the ROS? This is not yet known. So now this is the group what we have formed in BITS Pilani. So I'm an early career researcher. I have already formed my group. So they have two PhD students, and I have JRF, and our mission of our lab is to decode these sugary secrets. What is happening during a plant fungal pathogen interaction, to see what kind of glycans are produced during the plant fungal pathogen interaction, especially focusing on plant pathogens over here. So the plant pathogen what we work in our lab is the Macrophomina fasciolina, and which is a very important climate change relevant soil-borne fungal pathogen, because the pathogen virulence is dependent upon heat and drought. Whenever there is high drought conditions and heat, especially in the scenario of global warming, the pathogen virulence is highly high, and it kills it kills a plant by causing a classical disease which is called as the charcoal rot disease. And charcoal rot disease causes a yield loss about 70 to 100% yield loss, and the 70% 100 yield loss is a high, especially in important crops such as chickpeas, sorghum, and even soybean. So the model plant what we are using in the lab is the soybean. So soybean is an economically important oil seed crop, and also I have already told you that Macrophomina is a very important pathogen for soybean. So what Macrophomina does, it's a very important pathogen. So once it colonizes the root, and this is the colonization route what we have done in our lab on a soybean plant. So once it colonizes soybean plant, under a proper infection conditions like 2 dpi and with a high temperature, it forms microsclerotia in the intercellular space. So microsclerotia is nothing but the joined masses of a fungal hyphae, and these joined masses of fungal hyphae clogs the vascular system of a plant. Once it clogs the vascular system of a plant, the plant is going to get wilted because it's not going to get any nutrients. The plant will ultimately die. So over the different course of infection, you will see a lot of microsclerotia. This particular pathogen is going to form, ultimately killing the whole plant. You can see the visual symptoms of how a pathogen kills the whole plant in this particular image. The more important thing is that there is no reliable resistant gene sources or any resistant plants or resistant genes identified in crop plants to combat this particular pathogen. That necessitates an important mechanistic study in this particular pathogen. So my student Muthu, he has successfully isolated Macrophomina fasciolina, and therefore successfully isolated the extracellular polysaccharide matrix, which is produced by Macrophomina fasciolina, and he used the glycosyl, glycan profiling platform what we have established in our lab. Again, it was very interesting to see that the glycan profiles of the sugar profiles of the cell wall and EPS were completely different, or it is highly distinct, and it is also distinct from the beneficial fungi what I have shown you before. And the interesting thing is that, again, like beneficial fungi, we were able to see a very high concentration of 3,6 linked glucoses in the EPS matrix of Macrophomina fasciolina. This is indicating that indeed the EPS matrix of fasciolina has a very high amount of beta-1,3,1,6 glucans. We have done enzymatic assays and mass spectrometry to prove that beta-1,3,1,6 glucans are present in Macrophomina fasciolina. So apart from that, we are also able to see some interesting sugars in the pathogen cell wall, especially the galactofuranose form. So usually the sugars are present in the pyranose form. This is a pyranose form of a sugar, but we were able to see a furanose form of a sugar, which is detected in the Macrophomina fasciolina. We have already confirmed with the mass spectrometric analysis, and the relevance of furanose form is not yet studied, and we are continuing this in our lab. So we proceeded again with the beta 1316 glucans, and we try to ask ourselves what are the enzymes which are targeting the beta 1316 glucans in soybean when the pathogen is making this particular polymer in the plant. We took a soybean plant, we infected with the Macrophomina faeciolina, we collected the apoplastic fluids and performed the quantitative analysis, and we were very much amazed to see that we were able to see an upregulation or the a protein which is highly upregulated in the apoplastic fluids is a beta 13 endoglucanase, which is belonging to GH73, GH17 family, which is an exact ortholog of an enzyme in barley. So we have already shown that during an infection of barley with the Serendipita indica, which is a beneficial fungi, we were able to see a very high accumulation of GH17 enzyme. And again, in the same case, the same ortholog enzyme is also getting upregulated in the soybean as well. So what is the role of this particular enzyme? We are purified this particular enzyme. We treated with the cell wall. We were able to see a huge burst. That is, the cell wall is able to make a huge burst, or the fragments released from the cell wall is able to make a huge burst, whereas the fragment from the EPS matrix is not able to do the burst. And again, when we did, so we have an assay in our lab to know if there is any antioxidant potential of a digest from the coming from the EPS matrix. Again, we were able to see that with the chitohexose, you are able to see the huge burst. Whereas now, when you treat the chitohexose with the fragment which is released by the action of GH17 enzyme, you don't see any burst. Whereas now, when you denature the enzyme and when you do mix it with the chitohexose, the digest coming from the chitohexose and the GH17, you see the burst is coming back again. Again, this is indicating that the hydrolytic activity of the GH17 enzyme, which is an important factor to release the scavenging factor, which is coming from the EPS matrix. So we were very curious to know, is it the same fragment which is coming from the beneficial fungi, or is it something different? We did a glycosyl linkage analysis. We were amazed to see that we were able to see apart from 3 and 3,6 glucose, there is a very high amount of 6-linked glucose which is present in the EPS matrix of Macrophomina faeciolina. I've already shown you that this is a fragment which is released from the beneficial fungi. It has a 3 and the 3,6-linked glucoses only, and this is the fragment now. Now we are having a very high concentration of 6-linked glucoses. So we don't know why, what is the functional relevance of 6-linked glucose, but I've already told you that 6-linkage is relevant with the raw scavenging property. So now currently we are doing an NMR in our lab to know what is the structure of this particular fragment. But already we were able to see that the pathogen is again making use of the beta 16 glycosyl linkages to make the environment, to make it itself more adaptable to the environment, because it can survive in the high ROS conditions during the infection with the plants. So in our lab, we are also, I have already told you, Macrophomina faeciolina is a heat-sensitive pathogen. So it is also, very sensitive to heat. At high temperature, it can grow very fast. So we did a transcriptome analysis to see what are the changes which is happening at 28 and 35 degrees Celsius. We were able to see that several cell wall associated genes were actually upregulated at 35 degrees Celsius. That is a virulent condition for the pathogen infections. The cell wall. So we thought, okay, is there any cell wall remodeling going in this particular pathogen during at a very high temperature or the virulent conditions? Yes, indeed, we were able to see a very interesting fact that the 3,6 glucans, which was high at 28 degrees Celsius, was going down. The 3,6 glucose which was going down, and we were able to see a new polysaccharide which was coming up at the 35 degrees Celsius, and this particular polysaccharide is a beta 14 glucan. So beta 14 glucan we have already tested with the MALDI-TOF analysis, and we have also confirmed the beta 13 glucans. So currently we have also found a very interesting isoform of a beta 14 glucan. So this is the direction we are going, that the pathogen is having a beta 14 form of a glucan, which is different from beta 13 or 16. Okay, so with this, I would like to conclude my talk. So basically, I have told you my sweet journey, or told you about the different aspects of glycobiology which I've been working on. The main thing is that from mechanistic traits, we are going towards trait discovery, especially for the crop improvement. So I have told you about two important interactions. One is the plant-bacterial interactions and the plant-fungal interactions. The plant-bacterial interactions, I have shown you the important sugars, especially the glucnag and the viosamin sugar, and also immuno sugar, which is used by the plant to to manipulate the plant during infections. And I've also already shown you about the importance of beta 16 linkages in the beta glucan to scavenge the ROS, such that the pathogen or a beneficial fungi is able to grow very high in the plant. With this, I would like to stop my presentation with the acknowledgements. I'm highly grateful to my supervisors, especially Professor Rainier van der Hoen. Rainier van der Hoen is my PhD supervisor from at the University of Oxford. Because Rainier, my supervisor, Professor Rainier, he introduced me to the glycobiology world by giving the probes. If I would have not got the probes, to test, or if I have not tested the probes, I would have not gone to the into the glycobiology field at all. I am clearly say that. So I'm very grateful to my supervisor. Because of this, I came into my field. I'm also very grateful to my postdoc mentor, Professor Marcus Pauli, because most of the techniques I have learned from Professor Marcus Pauli's lab. I'm very grateful to that. And some of the part I have not shown over here about the plant cell wall studies, which I've done in Professor Marcus lab, and also Professor Alga Sukaru's lab, where I have done the extensive work on plant glycobiology, especially in the terms of plant glycan interactions. And the work which I have already shown you with the macrofumina faciliona work is done by my team. So we are as a oligosaccharide right now as a small team. I have a team of two PhD students who are hardcore in this topic, and we also have ME students doing their thesis, Shakshi, Vasudha, and Devansh, and also we have Naveen doing as a JRF. So currently we are as a oligosaccharide, and we are expecting to grow as a polysaccharide in this particular field, and also to provide our expertise and also extend our collaborations to Hodder. And also we will be liking to thank our collaborators from ISA Pune, and also Dinakar and Sidesh, who have been helpful for me to establish during my initial stage of establishing my lab, and also several publications. So with this, I would like to stop my talk, and I thank you all for your attention. If there will be any questions, or I'll be happy to answer the questions. Thank you so much.
Thank you, sir. Okay, Shubho. So, shall we start with the questions, or what? Shall I move forward with
Interview.
the interview
Interview.
section? Then you can collect the questions meanwhile.
Yes.
Okay, fine. So let's start this session by learning a little bit more about you. We know so much about your research journey, and so we would like to know a little bit more about you, sir. Could
Okay.
you please tell us about your scientific journey, and how you became interested in plant sciences, and what inspired you to pursue your career in plant microbe interaction?
Okay, that's a very nice question. So, especially with respect to plants, so I did my bachelor's in biotechnology in agricultural university. That is Tamil Nadu Agricultural University. So after my 12th grade, so I was having a passion to work with some crops, especially rice and rice yield. Something was very much fascinating to me. So with that passion, I also read about plant tissue culture at that time, that you can take a X plant, and then you can make a callus, and then the plant grows. So that is my curiosity, that how does it is happening, and there is a lot of application over there. So that prompted me to get an admission in Tamil Nadu Agricultural University, where most of our courses are related to agricultural biotechnology. We got exposed to a lot of things in agricultural biotechnology, especially right from abiotic stress and biotic stress. And I was fortunate to have my professor who was a plant pathologist, and he taught us plant pathogen interaction in a very interesting way about OR gene and virulence gene interactions. So that is my starting point to delve into plant pathogen interaction fields and plant sciences. And also, I was very fortunate to get a scholarship in plant science again in German government. So I think that is my thing, that I was in plant sciences and with plant pathogen interactions.
All right. So, yes, and you went to Germany after that. And how was your research experience there?
Your research experience in Germany, so I have to say it's more practical over there than theoretical. So when I went from bachelor's to master's in Germany, so it was like when I was in a master's course, students who were in the course, they were doing the experiments without from the instructors even. So instructors were
Okay.
giving a very brief instructions that, okay, this is what the experiment is, and start doing. So the students were very organized. They took the append of tubes. They were labeling it. They were doing all this in a very systematic way. So I realized that that kind of training I lacked during my
Mm,
bachelor's.
mm,
That though I was very strong in theory, I was lacking more practical side. So that is I learned. Okay, the practical is something I have to improve. So I improved that during my master's. That helped during my PhD and my postdoctoral during my postdoctoral work.
Okay. Now there's a lot of talk about AI.
Uh-huh.
How do you see AI transforming the landscape of scientific research and publishing in the next decade?
So, with respect to research, I can answer first. That is going to be a very important factor, I would say. Especially AI is being used in every part of research. I can see that AI is used in agriculture now to model or to predict something. AI is being used because we have huge data sets which is being generated over a lot of years. And how to make use of these data sets and how to make a hypothesis. So to make a realistic hypothesis, AI comes into the picture, I believe. So because when you have a large data set and if you want to make a hypothesis, it is going to take a lot of time. Rather, if you have a small data set, you can make a plan. But when you have a large data set and if you want to make a predictive and a very precise hypothesis, AI is going to play a major role, I believe, especially in terms of protein science, especially AlphaFold, for example. AlphaFold has predicted a lot of proteins, which is not possible to be even crystallized in the lab. So it's able to predict a protein structure. So having a protein structure prediction, you could now design your target of interest or drug of interest. So I think AI is going to be a very important game changer in any biological science field. And with respect to publishing, it's going to obviously, increase the impact factor of your research if you, include some AI and then show a predictive model, and then you have generated a wise hypothesis and you have proceeded your topic. Yes.
All right. And talking about the future, and so definitely the issue of food security comes into mind, and there's a concern going on. So, what are your thoughts on the genetically modified crops as a solution for enhancing nutritional content and addressing food security concerns?
Nutrition content and addressing food security with respect to gene editing.
Genetically modified crops
Yeah,
in particular.
genetically modified crops. So I can talk about genetically edited crops because GMO, genetic modification with respect to transgenic crops. So for example, that is going to be a very important, going to play a very important role because that is something where you have a new gene which is going to be in a system, and this is going to have a play a major role in either increasing the nutritional yield or the biotic stress or the abiotic stress tolerance, especially in terms of food security. That is what we needed. So right now, because of the government regulations, we are not allowed to have GMO in the field, but gene editing crops are getting a major hit right now. So already you might have heard about the two gene edited rice,
Yes.
which is already in the market. So that is a very important game changer, I think. Just changing one base pair and you are able to increase the panicle length and also number of grains per panicle. So that is highly amazing because we are limited with the land, and if you are able to make use of the whole land for such innovative things, I really appreciate gene editing and technologies in field.
All right. In talking about plant sciences, do you think that there are any particular specific field of plant sciences that is going to gain uh, popularity in the recent future?
Yeah, that is very hard to address. So I would say, every field has their own positive or more. How do you address is what something more. It's very important. I would say I will put the weightage in all the fields, which is so far going on in the plant science. But my gut feeling in one direction, which is going to be what I feel is that the gene editing, especially the gene editing one, which has shown a very promising thing in the field. And so that is something I feel like that is going to have let's say artificial intelligence combined with gene editing, so which we can predict, our candidate traits. Let's say you have a complex data, by using artificial intelligence, you come up with a trait, and then you're able to edit it very fast. So that is going to accelerate, application in plant
Absolutely.
sciences, I believe.
So are you currently also venturing into any new areas of plant sciences other than glycobiology?
Yes. So when I started my lab, we started with glycobiology, and obviously, like when you have students, and then when students are highly motivated and they are very curious, you try to, have some branches as well. So we have recently ventured into AI, especially artificial intelligence in protein prediction and protein-protein interaction predictions. So using AlphaFold multimer and AlphaFold, and so that is like the one which we are currently going in our lab.
And so when you are doing this research and guiding your students, so time management is a crucial thing that you have to do. So it is crucial for research and academics and also life. So are there any strategies or practices that you employ to optimize your time and productivity in your work and life?
Okay, so as a early career researcher, that is very important. So I have to also now balance teaching in our in our institute. So we do teach, and then we do research, and then we have work. So it's very important. The time management is something which I learned during my postdoctoral work. So I feel like starting your day early helps you to finish early. So when you start your research work or your official work early, very fast, I feel like I'm able to finish my work before 6 or 5, so that I can give some time to my family also. But this I have a very important thing, that I don't I have a time limit for a day, so that I'm balancing both work and life.
That's great. And so, what is how is your research going to contribute to the national biomanufacturing policy in the future? What do you think about that?
Okay, so especially biomanufacturing is something, Bio E3, I really like it, because it's like both biomanufacturing with respect to the applied side. So with respect to plant biology, glycans are very important for as an elicitor, which can stimulate as a, plant immune regulator. This can be scaled up, and this can be applied in a field. That is some important thing which glycobiology can be. And also some peptides can be also scaled up, and this can be also a boost to increase the immune system, and that is going to be a very important component as in the applied perspective.
And how do you approach to collaborations with researchers from different disciplines and institutes?
Yeah, different disciplines and institute. I have to say this is you have to take a step. I would say this is what I learned. For example, let's say when I started my lab, and when I need help, I mean, because like in every systems, you need to outsource something, or you need to collaborate and then get your work done. So for example, NMR analysis, if you want to do NMR with 800 megahertz, so we need to collaborate with someone. So I was amazed to see that when I write an email explaining my research, and when I am seeking for collaboration, so far I have not got any no answer. So, which means that everyone is waiting for someone to collaborate. So
Yes,
I think
yes.
that is something. So let's say when we have a back loop saying a negative feedback saying that, okay, no, it is very hard to collaborate. How do I approach someone? I think then then you don't get something. So I would say be very free. So you are going to get yes or no emails. So what I did to collaborate, so I write my research experience, and then I just approach saying that this is going to be very helpful. We are a biologist, and if I'm collaborating with a chemist, I say them that synthesizing this particular probes is going to have a huge impact with applications. Especially chemist people likes to synthesize, but they see application as well. So when you try to convince an interdisciplinary person by saying an application in biology or in the whole biological sciences, that gives a kind of a interest to a
Yes.
collaborating partner, and they come together and they, then the collaboration goes very well.
Mhm. Okay, so you are working with students, and so when you hire a person to work with you, or when somebody joins your team, so what qualities do you look for other than academic qualities?
So, other than academic, I see an intrinsic motivation is a very important factor. For example, like and a self-driven something. So, for example, when you give a keyword, a student should take the keyword and try to ruminate in their mind about the keyword, and that gives a passion for them to pursue in that particular thing. So, one I look for an intrinsic motivation, and other is like efficient time management. So for example, I have already seen that some of the students are very much able to manage your time and also plan the experiments very easily from their PhD studies onwards. So but I mean, I don't still I see that PhD is a learning curve, but I don't have that as a criteria because they learned this during the PhD. But the most important thing what I see is their intrinsic motivation on any topic what they are doing, because when they are going to join as a PhD student, because that is going to drive them for 4 years. Because I can give some motivation for some years maybe, but the next part is the one they are the one who is going to lead the particular project. So I see an intrinsic motivation is an important factor for a PhD student.
Yes, and one other thing about, an important thing about the PhD journey is publishing a paper, or and after PhD journey is manuscript writing. So, what advice would you give to young researchers about writing and publishing scientific manuscripts in plant science journals?
So writing and publishing is a part of a scientific journey. Actually, one learns actual writing during the PhD research, during the PhD studies only. So with respect to the, it also depends on the project, what you are working on. For example, like if you have a project and then you are able to, the project is going well from your first year onwards, you will be very lucky that you are able to publish this within the 2 years. It might be also possible that you have started a journey and it is something like it takes a lot of time to optimize something, and this optimization will lead to a very high impact factor journals as well. So in that case, what I would recommend is to start writing a review article, for example, review article of their topic itself, together with the PhD supervisor, going forward and putting forward saying that, okay, I'm interested in this particular field, can we discuss about different titles of this particular area in which we can review a particular chapter, so that gives a platform to learn writing during the early phases of PhD studies. Once this is learned, and once the student gets the research data, when it starts to flow during their third year and fourth year, this will be very helpful for them to publish their PhD work when they are in the final year. So I would say when the topic is in a such a way that it is like going to take some years to get a result, starting from a review and then going to a research article will be the best way, in my opinion.
So, do you have any words of advice for the students and scholars who are new to plant science research?
New to plant science research, read, I would say. So that is something which is it's that for example, my supervisor used to say me like this: a paper a day keeps your supervisor away. So he used to say this: a paper a day, like how you have a apple a day keeps a doctor away,
Yes.
a paper a day will keep your supervisor away. So you will be less dependent on your supervisor when you read just one paper a day. So in a week, if you are able to complete or read about relevant or something different, or participating in a journal club, and that will give you why you are in plant sciences, and then you will be enjoying this particular field.
That's actually great advice. Really good. So let's go back to Shubho and see if he has collected some questions related to today's talk.
Hi.
Hi.
Yes, I have collected, but most of the most of the structural related, which can be found from the Googling, or it can be found from the any biochemistry books. So we selected few of them, which is one is from your interview session, that scope of plant biotechnology or agriculture biotechnology. Is it here in India is better, or outside of India is better?
Okay. With respect to, I mean, scope, I mean, it all depends like how you define scope actually.
Agriculture, biotechnology, I think maybe crop sciences, I
Crop
think, or
sciences. Exactly. So I would say in India we have both, going in a very nice pace. Both basic biology and also the applied biology is going in both in a pace. Equally, I would say now in the current stage, when I look back before 10 years versus now, I see that it is more equivalent, both in abroad and also in India. So maybe in abroad you get more, you get more applied perspective more or in a more technical perspective. But research, even in India, in a very nice research institutes, you have an equivalent facility compared to the abroad ones as well.
yes, because I think every country has some of the elite institute and the research is top notch.
Mhm.
One question is related to their understanding. Maybe they want little bit elaborate about this step. What are the key steps in beta galactosidase or BGAL treatment for detecting ROS inducer, detecting the ROS in induced sensations and how it does to indicate ROS accumulations?
Yes.
Maybe some technique they are
Yes, I
asking.
think it's a very good question, also very interesting question. So the reaction involves HRP enzymes and luminol. So HRP is a horseradish peroxidase enzyme. Luminol is a reagent which can do a photoluminescence. But luminol will not photoluminescence unless reactive oxygen species is transferred to them by HRP enzyme. So HRP enzyme is the first enzyme which will perceive the ROS and it transfers the electrons to the luminol. So once it transfers the electrons to the luminol, luminol is going to get excited and luminol is going to get, after excitation, is going to fall to the ground state. When from the excited state, it is going to go to the ground state, it will emit luminescence, or you can say it's a photoluminescence, and this particular luminescence is recorded in the as a form of the ROS burst. So in other way, you can just say when you have more ROS, HRP is going to transfer more electrons to the luminol, and then you get more photoluminescence. So this is the correlation what you have in the technical part. So if at all your lab is establishing this particular approach, I would say to focus more on taking the correct HRP substrate and also correct luminol, and this assay works reproducibly and robustly well in most of the plant immunity labs.
Yes. So so far these two questions is we got
No, there
11
there were many questions and many appreciations in the YouTube chat. As you
appreciations is you can love it.
yes, you
Yeah,
will
lots
just
of
scroll and see it's amazing. And but there are many very basic
yes.
questions. Let
Now
us see the
we
Okay.
can discuss a little bit about here. Still 4 5 minutes at least. So as you mentioned that what what I understand that you are now establishing your lab here. So it is obviously very benefited to them. Also, those are the joining your lab to understand and this such type of experiment, which is
Mhm.
very not so common in here.
Mhm.
So you already published your paper in Science, I think, in two Science in
Yes,
2000,
yes, I was part
2019
of
and
that particular. Mhm.
2019 and 2025. So, do you feel that nowadays that those are the young PhD students, they are looking for the other stream than the coming to the PhD? Is it is it have you seen this? Because now many institute are going to face such type of issues like applicant number of PhD applications is reduced, not only the India, all over.
Uh,
So, what is your suggestions that how we can interact them to the research? So maybe the publications can be a reward, or maybe the interest.
yeah, so I feel that's interest is the main
Okay.
thing, because what happens is like you cannot force anyone to do a PhD. So that is it's not, a PhD is not like a kind of a, you think that okay, it's a kind of a job. You cannot think that like it's a kind of a training period which is going to make you to decide are you going to take an academia track, or are you going to take an industrial track or a entrepreneurial track. So, what I would say is that if a student is very interested with the entrepreneurial track, I
Mm,
would say UG as an undergraduate is the best age to try that because they get a lot of time to do some lot of hidden trial and then optimize many things, and then they will come to a point, okay, their product can be in the market. So that is a correct stage, I would say. So
mm,
masters is the second level to think, okay, are you interested with the PhD? And PhD involves a dedicated work on a particular topic. So are you really interested, and then how to perceive, and then having a passion for academic sector as well, because you during PhD also you will also engage in some teaching. So that should kind of passion should be also with the students to do the PhD, I
mm.
would say. So I think the intrinsic motivation should come from the student, and also if that is the case, I think if the basics are very clear, so that will motivate them to pursue PhD in any field of research what they are interested.
Yes. And few questions just came that is there any option to go abroad after finishing the 12th, means for the graduations? So when you go in the masters, I think you going abroad in masters. So have you feel that okay, graduations also can be done in Germany or anywhere?
You mean undergraduation?
Undergraduate. Yes.
Undergraduation. So, with respect to German system, undergraduation, you need to know German basically. So, if you are very fluent in German, and then because the whole studies of undergraduate is going to be not in English, but is going to be in German only. But some courses have been in plant sciences are very interesting. For example, you can see about CEPLAS. CEPLAS is called a CEPLAS. It's a very interesting cluster for plant science network in Germany, where this clusters have combined basic sciences and also applied sciences, and they're offering a very beautiful program on BS in quantitative science, quantitative biology. So this
Mm,
is a graduate, it's an undergraduate program where you will be exposed to a lot of statistics, which is used in plant biology, and also plant population studies, and especially in breeding and other things. So that is the one program which I feel which is relevant to plant sciences, and also BS degree in Germany, that is in offered by CEPLUS cluster. And obviously, I think in US or in UK, this is definitely possible because they are studies are in more in English. So I think this is possible, but the possibility for getting a for a funding might be less. But definitely, I think in English-speaking countries, this is possible to do a bachelor degree. But in German-speaking country, there is a very handful of programs where they offer a program fully in English language.
yes. And one questions we often to receive in bioengineering email also that how to communicate a scientist or mentor in case of before going to the abroad or selecting in a particular scholarship and all, you need some assurance for the mentor selection. So is there any technique you have learned there that okay, this is the way to how to communicate, or is it a random way? What is your
No,
take?
it's a very systematic way, because when a student he or she writes an email, it has been already it's very clear to someone who reads the email, is the student intrinsically interested in the topic or not. So when you take about the scenario in abroad, when you do a PhD research, they are very passionate about a topic what they have chosen for their PhD research. It's not that they have got the topic, they will work on this topic. No, it's not like that. So they are very passionate on this particular topic, and they are stepped in that. So that comes when you are more into a topic by reading a particular paper which a lab has published. Let's say you are interested in one lab, and then you are very interested to do a PhD in that particular lab. So what I would recommend is to read at least 5 papers which has been published recently in the particular lab, and what are the different aspects they are working on. So having this in mind, and when you go into that particular aspects and you start reading, you will come up with your own ideas. I bet you will come up with your own ideas. Oh, they have done this. Why not I try this particular thing? Why didn't they try this particular thing? And having this, when you write an email, you can approach a professor writing the same way, saying that you have read this particular papers and you find this particular research area which is not addressed, and you are very curious to address this particular research area. And then this is your CV and this is your cover letter. And with this email, the attention will go for a professor who is reading it or a PI who is reading it. Full attention will be on your email, and they take it serious that okay, you are serious in this particular position, and they can help you in if they have a funding which is from a grant, they can even give you on contract, or they can also recommend you for other fellowship programs as well.
Yes. So this should be a process, not the random, because nowadays writing a
Yeah.
mail is
Yes. For
yes.
example, if you don't address a professor's name, for example,
Mm,
dear doctor, professor,
mm,
or dear, if you just write a very simple
mm,
dear sir, it is common to all. So it gives an impression that you have copied and pasted for everyone. And
mm,
you can write, okay, this is what your experience and you are interested with PhD. Very general emails. This gives an impression that it's a very general email where you have not gone in depth. So if it is a very specific email, so then it goes or it gives an impression that it's a very specific email to this particular PI who is working on this particular topic, and then
mm.
the application is taken very seriously for the next steps.
Yes. So mm, thanks. Let Soma is complete these sessions. And Bala, I request you to stay 2 minutes after the ending. I just need to inform one of my research, but
Okay.
it's going to be not broadcasted. So that's why I
Okay.
need
Sure.
you 1 or 2 minutes.
Okay. Sure.
Soma, end the sessions and
Yes.
little bit give the idea how to they download certificate and all. Though everybody knows, but still,
I
I
am actually hosting it, so if you want, okay, fine.
no, it's okay.
Okay.
It's we already got 60 applications for the certificate. Now it's 68 applications. We received
So I will
1,000.
quickly just quickly
We
conclude.
yeah.
The students, the key takeaway message from sir, from what sir said is that you have to first read articles and read, and then if you want to impress your or you want to do good in your career, you have to read. And also if you want to apply or if you want to email somebody for a position, you have to read. So first you read about it and then you work on it. So and this session is being concluded now, and so if you are new to bioengine, so if you're new to bioengine, so you should know that you cannot just ask for a certificate at the end of a session. You have to be a registered participant. So if you have registered for this event prior to this day, then you can definitely apply for the certificate of participation, which you can do by the link that has been sent in the YouTube chat. Please click on the link and you will get the Google form and you can apply there. And please remember to use the same email address that you have used in your registration form. Two different email addresses, if they don't match, and then you won't get your certificate. So please remember to fill in the form very carefully and also put in the addresses wherever it is there. Okay. So that's all. And see you again next month in June. We have another session coming up. Thank you for all
Okay.
of you.
Thank you so much.
Yes.
So
Thank you.
Thanks everyone for
we'll
joining.
end the live streaming now.
Yes, thanks everyone for joining today's webinar. And every information is available in our website also. And we have already start receiving the applications, 120 applications we received for the certificate. So link is working. Many people are email me, and link is not available without any media, in any other media than this live
YouTube
chat.
chat. Yes.
Yes. So bye, everyone.
Okay, bye.
Thanks.
See you soon.
Thank
So
you. Thank you.
I'm going to
Please
end.
stop.